All right, hello. Yay, applause. Happy Wednesday. It's lovely to see such a good turnout when the weather has turned and now there are actually nice things to do outside. So thank you for showing up. I am Kirsten Nordstrom. I am a professor of physics at Mount Holyoke College, and I host and organize these events. If you have any suggestions for speakers, we are taking a hiatus for the summer, but I will be booking out for next fall and spring. So please send them my way. I'll be sending those invites out soon. So right now we're going to be learning about baby's first microbiome, how breast milk nourishes beneficial microbes in the gut. Our guest is Professor David Sela at UMass Amherst. I don't have too long of an introduction today, because like I said, this is our last event of the season, which is very sad. So I'm gonna leave it to David to discuss all of these things. These are the questions he's gonna be answering during the talk. This is a bit more about David. Again, he'll probably be explaining these things during his talk, so I'm gonna skip that as well. And just to say that we will be back in the fall, still here, still Wednesdays, so you can kind of do the math there. We'll send out a date announcement fairly soon, and then speakers will come later in the summer. You can go to scitechcafe.org and get on our mailing list. We're at Instagram, SciTech Cafe, Facebook, SciTech Cafe. All of that's all well and good. We are sponsored by the National Science Foundation, so please keep paying your taxes. That very, 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 very indirectly funds these sandwiches you're getting. So thank you for that. And more importantly, helps us do this. So this is definitely one of the imperatives of the NSF is to try to disseminate scientific information to the public. Woo! This is all the same jazz. If you have not signed in, please sign in over there. Midge has some wonderful tickets for you that might come in handy later. For anybody here for everybody here, is this your first SciTech event? Yeah, welcome, woo, welcome, okay. So those blue tickets will be handy later on, so hold on to them, don't lose them, must be present to win. I'll go over what you can win a little bit later, but there will be a Q&A after David gives his talk, and the blue tickets and the Q&A, they're all kind of related, so just hang on to everything, okay? If you need beer, you'll have to go outside to the other room. Please buy beer, or you can go through there, but you know, just be a little delicate with the audio and the door shutting and closing. I would like to thank the Ower and Brewer at Abandoned Building, Matt, who is somewhere wandering around, maybe in this room, maybe not, but thank you, Matt. The bartender, Deanna, please buy some beer and please tip her. Yes, as always. Like to thank Brian Nix on sound, Midge Harshorn and Lauren Engelbrecht for wonderful, wonderful, wonderful support here. This could not happen without them. And so I don't forget to announce this, please do bust your tables afterwards. Glassware in there, there's a trash can there. I will say that again later because I don't work here. And we are basically bringing this trash in here and consuming it. So please let's do our part to clean it up. And without further ado, I'm going to hand over the presentation to our guest speaker. Let's welcome him. And we need to switch over the presentation. So give us 1 second. Okay, wonderful.
Good evening. You can hear me okay, right? Hello, Dr. McClemens, I see you back there. Hello, Crowdervilles. Hello, Mom and Dad in Florida. Thank you so much for joining me today to talk about my favorite thing, which is about our research. And we're gonna talk about babies, we're gonna talk about the microbiome, and we're gonna talk about other aspects related to those two topics too. And so I'm seeing in the back, if my voice is not carrying, please just raise your hand and say speak up, or just do that. You can't hear me? Julian, you are very funny. Okay, so as I understand it, we're gonna have questions and answers at the end of this presentation, but this is what I'd like to do. If you have a question in the middle of my talk, and I don't know if this complicates things too much for Kristen, we could take notes, okay? And I'll also ask you, as several times, to just shout out an answer, very quick, right? So we won't need a mic for that. All right, so how can I make this about me a little bit more now that you're staring at me and I have the big fancy mic and the loud voice in this room right now. That's me up there. That's me over here in front of you. As Kristen said, I'm an associate professor at UMass Amherst, and these are all the things my laboratory studies: nutrition, human milk, the microbiome, cannabinoids, probiotics, and genomics. This is all the fancy places I've been to in my career. Happy to talk to you about that later on if you'd like. You are not here to see me talk about that or hear me talk about that. You're here to talk about what we've learned. My lab studies how what we eat impacts our experience. And when I say our experience, I mean our health, the way we go about our business every single day, the way we feel about ourselves, and other aspects of our experience, and we'll get to that later on in the presentation too. And we see it's interconnected with a couple of very important topics. We eat food. This individual eats food. And it's all interconnected with our health and the gut microbiome. And so let me see by a show of hands, who here has heard of the gut microbiome? Loud and proud, I love it. Very good. If I asked this question 10 years ago, I'd probably see maybe a third of you raising your hands, and I appreciate that. This here is my lab group. I'm standing there in the middle, and these are the folks who do the science in my laboratory at University of Massachusetts. I'm not only a professor, but I'm also a research scientist. And so for 12 months out of the year, I do science, I do research. And then 9 months of the year, I actually teach classes, and which is great. I love teaching classes. I like doing things like this as well. So now we're going to get to the human gut microbiome. What exactly do I mean when I say the human gut microbiome? And so I'm going to give you a brief overview of what that means. It means a lot of different things to many people, and we're going to briefly define it. Of course, as a scientific talk, so we're going to get on the same page about this stuff. As you might be aware, there's a lot of interest in what is known as the human gut microbiome. There's a lot of books written about it. There's a lot of podcasts about it. There are people buying probiotics off of Amazon, looking to enhance their microbiome on a daily basis. And this is only a small subset of those opportunities one can purchase to understand more about their microbiome and how they could enhance health. And maybe some of you have read these books. I'm very fortunate. I've been in this field now for 20 years, and a lot of these books, well, actually most of these books I have not read, but I know the authors, and they're great. If I could recommend one, it's this one over here. And some of you might know Ed Young from The Atlantic, and he does a great job. He did a great job during the pandemic, and he's still doing a great job. So what is the human gut microbiome? Well, microbes cover every single surface of our body. That means on top of our skin, as well as inside of us. And so inside of us, in the gastrointestinal tract, is actually an external surface. You think about it as inside, but I think about it as an outside, because when we are developing in utero inside our mothers, we start off as one cell, which becomes 2 cells, which becomes 4 cells, 8 cells, 16 cells, and so forth, right? And eventually you become a little mini pancake that folds over onto itself. If I didn't have a mic, I'd do a better job demonstrating that right now. And so what ends up becoming your gut or your gastrointestinal tract actually is an external surface. And every single thing that we consume, all the food we eat, the air that we might swallow by mistake, edibles that we might consume, the beer we're drinking, all that stuff is touching upon that external surface of the gastrointestinal tract, and that's a main way by which our bodies communicate with the outside world. Microbes inside our bodies, in the microbiome, outnumber our own cells. And that was surprising for me to learn about 20 years ago when I started this, this scientific process or started my training. I didn't know that before. I knew that there was microbes inside of us. And when I say microbes, I'm talking about small, tiny, living organisms that you can't see unless you look at a microscope. And when you go to the bathroom, you're aware of you have to wash your hands because you don't want to get microbes all over the place. Of course, that's well known to us in this room. But knowing that we have a lot of microbes in us, that is actually relatively new from our scientific understanding. Now, if we wanted to get rid of those microbes, we couldn't do it. You could take all the antibiotics you want. It doesn't matter. They'll come back sooner than later, right? So people who are infected with something and need to take antibiotics, you actually don't sterilize your body. They eventually come back. Now, our microbiome provides several benefits. It provides benefits to our immune system, our metabolism, our psychological health, and also helps out with our development. Each location of our body hosts a different ecosystem. And what I mean by ecosystem is like when you look at a forest, that's an ecosystem. When you look at a forest, you see lots of furry animals, you see trees and grasses. All those animals and those plants and those grasses, they all live together and they get along or they reach a balance. And that's an ecosystem. And so when we're talking about a microbial ecosystem, I have an ecosystem behind my ear. Dr. McClements, all the way back there, has a different ecosystem behind his ears, but they happen to do the same thing, right? I have a different ecosystem than Maggie does in her guts, but they do the same thing if we happen to be relatively healthy. And we've evolved with these microbes over millions of years, and they're here to stay. So we don't want to get rid of them. They do beneficial things, and that's what I mean when I say the human gut microbiome. You've seen this picture before, and maybe in your high school biology class. This is a food web. I'm not going to spend too much time on it, but you see those furry creatures I was talking about, and the trees and the grasses, all that kind of stuff. But it gets a little bit more complicated when we look inside our guts. It looks like this. You don't actually, and I'm not going to spend too much time on that. I just want to give you a broad overview of what we do in a laboratory. All these fancy names over here, don't worry about that. Those are all different microbes. And we know about this in our laboratory, and they all have these interconnections, and it gets very complicated. And so as scientists, what we try to do is reduce the complexity. So what are the important key actors in these pathways over here? How do we influence the gut microbiome? If I say the gut microbiome is essential for immune health, what can I do as a scientist interested in advancing our species by perhaps modifying the microbiome by making more of this in my gut? Can I recommend a diet? Can I prescribe a probiotic that might help out what's going on over here too? So the take-home message here is that we learned a lot in biology about what's going on here in these ecosystems, and we're using the same sort of approach with what's happening in our gut, except we can't see it. We can see what's going on in the forest, but we can't see what's happening in the gut. All right, another key set of concepts here is that human ecosystems are diverse, and we have the most amount of diversity in the human gut. About 800 different species in the mouth, about 500 in the vaginal tract, 200 in skin, 100. It's give or take, depending on the person, depending on the time of day, that sort of stuff. But inside of you, you have about 800 to 1,000 different species of bacteria that impact your health and your wellness. One question that I always get is there a healthy microbiome? Is there such a thing as a healthy microbiome? Well, my question for you folks, and this is, is this a microphone opportunity? Is this something we can pass out to the crowd? I love asking this question. This question is actually a much more difficult question than you might seem on the surface, is defining what is health. So what does health mean when I say that word? What is health? Who would like to raise their hand and answer that question? I'll call on you, by the way, especially if you don't make eye contact.
All right, I'm going to bring the mic over.
We have a brave soul over there. That's table 8. The opposite being sick. That's a great answer. Thank you. I'm going to get back to that. I appreciate that. I appreciate you for raising your hand. How about another answer? Yes. I'll say that's table 11.
Like physical fitness?
Physical fitness. I appreciate that. I see a hand in the, well.
Oh, there we go. Thank you.
Yeah, so isn't health really just a state that you're in at the time? Because you can have good health or poor health, and it's kind of like taking, a picture in time.
Absolutely. So you're taking a snapshot, and I appreciate the way we measure health. How about one more answer to that?
Okay, I got you.
I think it was Jason, right?
Yeah. I would say good health is the state where you don't have to worry about your health.
Okay, well, I appreciate that. These are all great answers. And by the way, I always ask my class what this exact question, and I'm so fascinated by the answers. And the first answer, the one that you gave, I appreciate that, is the way the National Institutes of Health defines health, primarily the absence of disease. So if I don't have cancer, if I don't have cardiovascular disease, that means I am healthy. And we also had, I believe, an answer about performance, if I am physically fit, something like that. There was another answer about maybe how we measure health. It's a snapshot in time, how we are feeling in this moment as well. And then I'm sorry, I forgot your answer already. It just happened. Well, you don't have to worry about it too. So it is actually your perspective or your perceptions of health. So am I healthy? Am I not healthy too? And these are all moving targets. Oh, there's a hand over here.
Thank you. Eliza says being healthy is not having someone else's germs.
Oh, I appreciate that as a microbiologist. I am a trained microbiologist, and so not having someone else's germs, I buy into that. Thank you, Eliza, for that one. These are all appropriate answers, and what I'm trying to get at with this is that there's not one way of defining health, even though we use that term very loosely every single day. We always talk about, I want to eat healthy, I'm feeling healthy, I'm not feeling healthy, and we're not always thinking about it. The National Institutes of Health is defining it, as I said, as the absence of disease, but we in our laboratory are taking a look at health from another standpoint. We want to look at towards performance, which is another aspect. We want to eradicate diseases, we want to address diseases, we also want to enhance human performance, which means, how can I give a talk and not stumble on my words? How can I jump higher if I'm playing a pickup game of basketball? And those are things that maybe I don't need every single day. But we're talking about when you're looking at an Olympic athlete, for example. And oftentimes when I give this talk, I give an example of Olympic athletes and their various body types and forms, and they have unique needs, nutrition needs and unique needs. Their microbiomes have unique needs and so forth here. And all these things factor into health. So it's not only just the absence of disease, even though that's a big part of it. It's not only just making sure you don't have other people's germs as well. And so defining what a healthy microbiome is, or what a healthy nutritional status or diet is, actually a very complicated topic for scientists. We have another complication over here. And I see a couple of fast readers over, you guys skipped ahead, I love it. So differences in microbiomes often correlates with diseases. Often correlates, meaning that I can take 100 individuals who have Crohn's disease and 100 individuals who are healthy, and I can say, oh, their microbiomes are different in the Crohn's disease. But that's a correlation, and that's a problem, because it doesn't necessarily imply causation. We don't know if their microbiomes are different because of Crohn's disease, or their microbiomes caused Crohn's disease, or it might be what's known as a spurious correlation, where the two things don't actually matter with each other. And I have an example here of the divorce rate in Maine. This red line over here is decreasing at the same rate as the per capita consumption of margarine. Do we think there's an actual link between the two, divorce rate in Maine and the consumption of margarine? The data over here, that's an R value of 0.99. It's a very clear correlation, but a lot of times in science, we get these correlations and they have nothing to do with anything that's important, which doesn't mean that science is BS. It doesn't mean that science, well, I'll speak for myself, right? It doesn't mean that there's a problem with the way that we go about things. It's just, it's a limitation. Science cannot explain everything because we are limited by a couple things: the tools that we have, the human observer, myself, the people in my laboratory, the availability of samples and stuff like that too. So the microbiome field is very young. We don't know everything that we're going to know in 10 years from now. And that's exciting. That's exciting for me. That's exciting to the graduate students in my laboratory. That's exciting to my collaborator, Dr. Julian McClements back there, who's excited for this kind of stuff. Here's another example here. I like these ones. Per capita cheese consumption in red. Number of people who died by becoming tangled in their bed sheets. Look at that clear correlation. I could write a Nature paper on that. Nature is a fancy scientific journal that we get a lot of credit and kudos at our institutions. We go there. I'm just kidding around about that. This is what's known as a spurious correlation. This is not necessarily a clear linkage here, but you can massage your data enough to end up, it ends up looking like this. The take-home message here is that the microbiome and understanding the microbiome, again, is still in its, dare I say, infancy. And we're still trying to learn what's a real correlation and what's something that's not a real correlation, and how we can leverage that information so that we have some real world outputs. Okay, well, what we do know about a healthy microbiome in general is that diversity in the gut microbiome makes it more resilient to diseases. So if you have a diverse set of microbes in your gut, you're most likely going to be able to withstand any sort of microbial pathogenic, pathogens are germs that cause diseases, infections. You may or may not be able to, but you have a better chance to do so. It's also difficult to introduce new microbial species such as probiotics, but it's not impossible. So when you purchase that Akkermansia from Amazon, you end up introducing that probiotic to your gut, but it doesn't stick around. So that's why you have to take one pill a day so that you maintain those levels at the appropriate levels in order for it to do something. There are external factors that could have a large impact on the various microbes in your microbiome. And it's all about the balance. That's why this elephant's balancing this ball over here. And going back to the diversity over here, it's the same concept as if you have a monoculture in a field. If you're just growing corn, it's more susceptible to having weeds grow in your cornfield. But if you have a large overgrown garden, you're very likely not going to have as many weeds grow that are introduced as new species to it. So over here, external factors. What are some external factors you think could impact the gut microbiome? The gut microbiome are the beneficial microbes in our guts, right? What do you think impacts our gut microbiome? That's a question for the audience. We'll go diet. Diet. What else? Infection. I'm repeating this for the people at home. I know mom and dad are there. Stress. That's a very good one. Stress. Was that?
Mental health.
Mental health.
Mental health. Absolutely. Absolutely.
You wanna mic it?
Medications?
Medications, the things that you take over the counter or prescription, things that are introduced to your body, these are all external factors, including antibiotics, right? Thank you, I appreciate that. So the external factor that I anticipated as a nutrition scientist that I study and most concerned about is diet. And that's how one can modify microbiome function, form and function, by eating differently. And there's a lot of studies out there, some very, very strong studies, that if you go from a strictly carnivorous diet to a primary plant-based diet, that you can change your microbiome's function rather rapidly. And a lot of these studies were done in animals, but some of them have been done in humans, and they're reproducible. But of course, I'm not talking about adults, we're going to be talking about babies' microbiomes. And so what do babies eat? You could just shout it out. Milk, that's right. They drink breast milk. Babies drink breast milk. And that's the reason why I'm in nutrition. I asked myself, so I originally went to UC Davis to study wine microbiology. And that was a lot of fun. But there are some things that are better left as a hobby than as a career. And then there was a couple of projects that came underway about breast milk. I was like, breast milk. I never really thought about studying breast milk until I started realizing that this is the one food that specifically has evolved to benefit our species. There's no other food like that, right? It's the only food that's specifically for our species. And so I wanted to understand nutrition at its fundamental basis. This is my friend Bree over here, who is, her baby is now 2 years old. This picture was taken a few years ago. Babies don't only eat milk, right? Babies get into stuff. I'm gonna swear right now, but babies get into shit, right? Like there's some stuff that babies get into, and that all contributes to microbial health, and it has a lot to do with what microbes they have inside their bodies. When you handle a baby, you're passing microbes to them, not in a negative way. That's how your baby's body is learning. right? And so we're starting to understand what is the right level of hygiene when handling a baby. Do we want to always use the hand sanitizer? And by the way, I'm not recommending anything. I'm not saying you should or you should not do it. You do whatever you need to do, right? Because we actually don't know. But babies have traditionally evolved to be messy. And that's over thousands and thousands of years. Okay, so how does breast milk feed babies and their microbiome? On the left, we have what's in formula milk. On the right, we have what's in breast milk. You see a lot more goodies in breast milk, which does not necessarily make it a better thing. It certainly does not make it a worse thing. It's just different. And I'm used to talking about this topic, not only about breast milk, but when I'm lecturing nutrition students at UMass Amherst, we always talk about the values we put onto foods by calling foods good versus bad. And so I'm not saying formula is bad and breast milk is good. I'm saying that there are different things. And it's the same thing with many other foods. If a food is inherently bad, it's probably not a food. It's a toxin, right? And of course, there is a dosage effect because a sip of cola is not going to be bad for you. But if you drink 3 liters, an hour, you're going to have some challenges, right? So how does breast milk feed babies and their microbiome? Well, infants don't digest these complicated sounding things that I've been looking at for 20 years, so they don't sound complicated to me. They're called human milk oligosaccharides. Human milk oligosaccharides. I'm going to call them HMOs from now on. There are about 120 known structures. Don't worry about understanding them all. Most of science doesn't really understand them. We don't fully understand them as scientists in this field, but there's a lot of diversity of HMOs that are secreted in breast milk. And actually, there's a lot of HMOs in breast milk. There's actually more HMOs than protein in breast milk. And you think about milk, we're thinking about cow's milk. And we actually know more about cow's milk than we know about breast milk. And there's more protein, or there's more HMOs than there is protein. And so as a graduate student, I was curious about this. What exactly is going on with HMOs? Why is there so much there? And as it turns out, they're indigestible. They're not digested by infants, but guess what digests the HMOs? You could shout it out. Microbes. That's right. You are a distinguished audience. I appreciate that. I appreciate you. Bless you. Bifidobacteria over here. This is our favorite bug that we study in the laboratory. When I say bug, I'm talking about microbes. I learned very often or early in my career when I'm giving lectures, I go back and forth between microbe and bug, and I had a student raise their hands like, what are you talking about bugs? I thought this was a class of microbiology. What are bugs have anything to do with it? They thought I was talking about little crawly things, right? So that's not what I'm talking about. These are the bugs I'm talking about. These are bifidobacteria. I spent 20 years staring at these things over here, these like Y-shaped things. They look like antibodies a little bit. They look like Ys. These are bifidobacteria in the baby's guts. And if you're a breastfed infant, you have a lot of them. If you're formula fed, you have less of them. Again, we don't really know fully why bifidobacteria are better for babies, but we have some ideas. And we have some examples over here. So bifidobacteria inhibit diseases. I think Eliza was helping us out with, we don't want to catch other people's germs, right? They inhibit diseases by taking up, this is a fancy scientific term, by taking up real estate in your gut so that other bugs can't cause infections. They limit viral infections as well. There's metabolic benefits with bifidobacteria. They reinforce the tight junctions between cells in the gut, which is a bunch of mumbo jumbo about how when we have bifidobacteria in our guts, the cells, our own cells, our human cells, are tightly packed. When we don't have bifidobacteria, they're more leaky. So you get all kinds of stuff goes into circulation when we don't have bifidobacteria. They're anti-inflammatory, and they produce all these beneficial chemicals or substances that keeps the baby healthy. And we also have them as adults too. So the take-home message over here from this slide is that bifidobacteria are positive. And now HMOs are incorporated into infant formula. So as I mentioned, I alluded to this, that we started this research. And when I say we, it's more of the, is it the royal we? It's a large field. It's not just me. It's not just my laboratory. There's hundreds of scientists around the world who study this sort of stuff here. But when we started trying to understand HMOs and what that has to do with the baby's microbiome nourishment, we didn't know there was going to be an application at the end. We kind of thought, oh, maybe we could improve infant formula. But as it turns out, there was a lot of excitement and enthusiasm from formula makers for this, and they've improved their infant formulas by putting HMOs in them to make the formulas more human-like. Okay, so HMOs are a special type of sugar, and I forgot to mention that. They're just not digested by us. They're a carbohydrate, so you guys know what carbs are. But what about human milk nitrogen? As it turns out, there's a lot of urea in breast milk. Who here knows what urea is? You want to shout it out? It's in urine. That's right. Urea is in urine. That's why urine is called urea. That's why that's why urine is referred to as such. It's got a lot of urea in it. It's an excretory product. It's something that our body is trying to get rid of, right? And so we asked ourselves this question a couple of years ago: why is there so much urea in breast milk? Essentially, and I don't want to put it in crass terms because the nursing endeavor, the breastfeeding endeavor, is such a big part of how our species has propagated over thousands and millions of years, and people look at it oftentimes through a sacred lens. But why essentially is there pee inside breast milk, right? And so we had a hypothesis about that bifidobacteria use the urea that's typically peed out or put into breast milk as a nitrogen source to produce useful substances for the baby. And you don't have to worry too much about what's going on over here. This is that PowerPoint crutch, as they say, something for me to point at while I'm giving my fancy talk over here. That human milk comes in here. This is the baby's gut. This is the bifidobacteria. It consumes the urea and it spits out beneficial stuff for the baby. And as it turns out, thank you, NIH. NIH funded this research. That is appropriate. I appreciate that too. That's funded this research about 2 years ago. And that made me and my laboratory very happy because we were able to do this research. And so in a certain sense, I graduated, right? That's me a couple of days ago at UMass graduation. And then we actually had to do the science. And as it turns out, this is a recent press release, that we found out that bifidobacteria actually do utilize this urea, utilize this excretory product that ordinarily is just flushed down the toilet, and is used to make useful things for the baby. This made us really happy, so much so that Sam, the Minuteman, he got excited about this too. And with that, I'm going to allow you to ask any questions you might have about the microbiome, probiotics, urea, human milk oligosaccharides, and so forth. So thank you very much for your attention. Mom and Dad, I love you. That was so cheap, by the way. I did that so the questions would be easy. I'm just kidding.
Okay, so we'll take questions from the audience. There will be a prize for best question, which is sometimes the worst question. So it depends on what the speaker thinks is the best question. So I'm going to be passing around the mic to anybody who has a question. Okay, we'll go here.
So your slide, like 3 slides back, had nickel in it. I normally think of nickel as being kind of toxic, and I could be way wrong about that, but what's going on with the nickel?
Well, Keen, I appreciate you calling that out there. So nickel in high concentrations is toxic, but there are certain enzymes that use nickel as a cofactor. So in this instance, this enzyme, or this protein that makes this chemical reaction go forward, needs nickel to do that. So it's just a very small amount of it. And the same thing with other metals in the human body as well. But that brings up the point that there's a fine line between something that's a toxin or something that's beneficial for you. So too much nickel, not helpful. Too little nickel, not helpful. There's the Goldilocks moment.
Okay, and you?
Yeah. So I was wondering, with your research, did you find out that the same substances would be useful to adults, or is it just primarily in the infant stage?
Oh, I appreciate that. So we do what's known as fundamental research with an eye towards translation. So we're not actually making products, right? So when I show the formula, that's other people who are doing that. However, we're always looking towards it. HMOs specifically, there are people who are very interested to see how they could enhance adult health. with HMOs. And that kind of research, I'm seeing research come online in the last 2 years where people are looking to cure things like traveler's diarrhea or maybe other sort of concerns that people have with their gastrointestinal tract. So that's a question that's been coming up in the field for a long time, and we're starting to get answers.
Follow-up question on that?
Absolutely.
So with the urea as well, because I've seen some supplements that have that in there. As an adult, is that something you should be taking?
I appreciate that question. So with urea, that's brand new research. We don't even know the importance to the baby just yet. I think it might be a secondary way by which the baby reclaims lost nitrogen in certain contexts, but we don't know those contexts just yet. So even for adults, we're not sure that's going to be helpful yet, which is my scientific way of saying, like, let's pump the brakes. There's always going to be a tension between clinicians or people who are practitioners who say, okay, we need something quick, so let's go forward. And then there's going to be scientists who are going to be more cautious about that to say, hold on, we're not quite sure yet. But if you listen to just scientists or just clinicians, it's not going to be as helpful as reaching that balance. I appreciate that.
About 20 to 25% of patients will get antibiotics in labor, and I'm just wondering do we know how long it takes babies to recover, or if recovery is needed?
It's a really good question, and that dovetails with oftentimes the question is vaginal delivery versus C-section also. So you have a different chance of inoculating the baby or introducing microbes to the baby during delivery, and it changes with antibiotics. With antibiotics, we know that it's different. We don't know if it's good or bad, it's just different. And there's people who are looking at reintroducing mom's own microbes to the baby once, in the next couple of days while they're still in the clinic. There's various names for that. The fecal. So which one are you?
Wait, we have somebody who knows.
Vaginal seeding?
Yes. I thought you were getting at fecal microbiome transplant for some reason. I was confusing that too. Thank you for putting it out there.
Okay,
Vaginal seeding.
I see a hand here. I'm coming over.
So one of the advantages, I assume, of breast milk is that it's easily digested by the baby and it's easy for their gut to eliminate. So how old does a baby have to be able to successfully digest other foods?
How old does a baby have to be? So when we take a look at easily digestible, it's typically from our own experience, right? The baby does tell us, of course, when they're not having a good time with it, and it depends. So the recommendations, we have WHO recommendations for 6 months of breastfeeding, which, by the way, is being extended. I don't know if you're seeing the same thing. I think you're asking, you might have different, but it depends. Oh, I see what.
So the question is, how drastically does the microbiome change when you introduce new foods?
Right, okay, I'm sorry, I didn't catch that question. So as soon as you introduce solid foods like rice milk or something like that, something that's a little, that's introduced either here we find in so-called developed countries or maybe in indigenous cultures also, they have different food regimes as well. You see a shift and it's more chaotic for 2 weeks once you introduce that solid food or solid-like food to it, and it becomes more of an adult-like configuration, which is designed, well, if I could use the word design, it's more likely to include more protein in that diet too. So the function of the microbiome changes rather rapidly as soon as you add anything back to the baby's diet that's aside from breast milk, if that makes sense.
Oh, great. We have a couple hands, and they're in the same spots. That's very handy for me.
One of the press releases we saw up there said that your research is or could improve infant health. Could you describe how it can do that either now or down the line?
Absolutely. So that's the urea work, and I appreciate you asking that question about improving infant health. That if this happens to be, so in breast milk we have adequate protein, that we find adequate protein, and actually even if mom's undernourished, you're still going to get adequate protein in breast milk, right? But in certain instances, you might actually need more nitrogen. We don't know yet what that might be, but our laboratory and of course the National Institutes of Health and the people we collaborate with, we think that this might be a backup mechanism when there's not enough nitrogen within breast milk too. If that, does that? Yeah, so there is a possibility of modifying formula, but as we were talking about nickel, urea is actually a toxin. Your body's trying to get rid of it too. So the National Institute of Health is trying to understand fundamental roles of these processes so that maybe that there might be an application down the line where in some instances where there may be undernourishment. This might be not relevant for where we're sitting here in East Hampton for most of us, perhaps in our peer group, but it might be for other populations. So it's really dealing with nitrogen balance within the infant gut.
All right, we have one here.
You had a chart that showed the difference between formula and breast milk as far as ingredients goes.
Yeah.
Where would biomilk fit on that?
Biomilk. I'm not sure I'm familiar with what biomilk is.
It's lab cultured breast milk.
Oh, okay, lab culture, yes, absolutely. So, and I appreciate that. They're sitting 2 doors from you right there as someone who's very interested in cultured meat or alternative proteins. And there are a few startups that are based in the United States and one's in Singapore that's looking to make cultured meat technology available to make breast milk in these big vats, like very similar to those over there, like the ones that you brew beer in. And right now, the problem is scalability to make it commercially available. So you can do it in a laboratory, and it's pretty good in terms of replicating what's in breast milk, but making it scalable where it doesn't cost $5,000, you know, for this much. That's what the challenge is. Yes.
Does biomilk have the same set of ingredients?
Yes.
Is that correct? Okay.
Yes, it does. So it's using, a lot of these processes are using stem cells that will be able to secrete the same sort of stuff that you find in breast milk.
All right, we have a question here.
Hi, you mentioned earlier about how diet can influence microbiome, and it made me wonder about how different or consistent is it around the globe. So if you have mothers' diets, plant-based, carnivore-based, whichever, are they providing through their breast milk a slightly different population of flora and fauna in the gut than might be found in some other country where the primary diet is lima beans as opposed to, you know, some fancy other something else? Just curious.
Yeah, I appreciate it. So looking at geographic,
Okay, so the question I think is globally there are dietary differences. So you might have a more plant-based diet or a more varied diet. Will that affect the baby's microbiome? Yeah, more or less.
Yep. So the differences in breast milk between individuals who are lactating across geographies or so across the world, different cultural practices, right? Different genetics. That's an area of active research. And there's no good set of rules that we have right now, right? But we do know that certain foods, and those of you who have lactated before know that certain foods might make it taste different or make it smell different, or maybe look different too. So we do know that there are differences there. When it comes to things like HMOs that are secreted in breast milk, there's 120 different structures, but each woman's going to be making, or someone who's lactating is going to be making 50 or 60 different kinds. And there's an overlap, there's a big overlap, but we don't really know exactly what goes into it. We don't know the genetics that happen to say this set of HMOs are going to be from this individual secreting milk. And so that's a very, very good question. There's a conference next week that's going to be the exact topic of it.
I'm going across the room.
Eliza's question was really similar to that one. She remembers milk sometimes tasting different on one day from another day, and she was wondering if that means that there were different things in the milk that she was getting in her body. And Lily's question is, why only girls make milk?
Well, let's tackle the first one. So Eliza, yes, absolutely. That there are different chemicals that your mom had inside of her when she was eating foods, and you can taste the differences in breast milk. In some instances, it's not only girls who can lactate. There are instances in which people who identify as other genders or other sexes do lactate as well. So it's not only just girls. Thank you for those questions.
Okay, so we actually have two here. Can I?
Is there a significant chance or any chance that the urea in the breast milk is just a quirk and not actually advantageous?
I love this question because we always have to ask ourselves this question, is it just there? And it's just there, it's some sort of, and when we were doing the promo yesterday on the radio, it came up about, well, if it's there, didn't evolution make it there? And evolution is ideal. And actually, it might have been useful in the evolution of our species, but just a carryover from another time. And it just happens to be there right now. Or maybe it was never useful. So evolution is not necessarily something that's optimal or ideal. And there's lots of things that we can point to why that might be. I said yesterday about one's appendix. Maybe it doesn't have a function currently, but it used to have a function. How about things like anxiety? So usually we're running, anxiety, and the classic example is, well, we're not necessarily in East Hampton running away from tigers chasing us right now, but if I get an email from my department head says, I need you to do this tomorrow, it feels like a tiger is chasing me, right? And so things that have evolved to help our species may not necessarily be useful in this current context. So it's not only just the current set of genetics that we have right now, it's the context as well. And that's a really good fundamental question we have to ask ourselves. Just because it's there, is it useful for a function? And so the first step was to exclude the function that's feeding bifidobacteria. And right now we can't exclude that because we have, I didn't show really the data, but we see that it actually feeds the bifidobacteria as nitrogen. Whether or not that's important for a function that could give us an application with the question that was asked before, that's left to be determined.
Okay, we have another one at this table.
Are HMOs susceptible or resistant to changes in temperature, such as freezing or scalding?
Oh, that's a good question. I appreciate that. So, so you're talking about expressed milk when it's. So the way I take a look, the way I look at milk, it ceases to be milk as soon as it touches the baby's mouth, right? It becomes digestive because you have enzymes in the baby's mouth. When we're storing milk, that's a relatively new innovation in our species, right? And so within the milk itself, there's all kinds of proteins and enzymes that can change the formulation of the milk itself, including bacteria within the milk. So when you keep it out on a counter, it's going to change over time more rapidly than when you put it in the fridge or the freezer. And the HMOs are resistant to that, but only to a certain extent. Now, if you boil it, you said you scald it, you might actually have certain types of HMOs that are a little bit more sensitive to that too. And that's something that people who are studying, well, which ones should we put back into formula? They have to ask themselves that question. Which ones are actually going to survive that process? That's a really good application-based question.
We have time for maybe one or two more questions, and we have one here.
I think you mentioned early on that our microbiome can affect our psychological state. I'm just wondering if there have been any concrete examples of that in the literature of a certain bacterium or set of bacteria that are associated with, I don't know, depression, anxiety, or some other sort of issue like that.
Yeah, I appreciate this question because our laboratory does study this as well. And so I talked a lot about bifidobacteria, but what we want to take a look, so we take a look at bifidobacteria as a simplified proxy for the total microbiome. We have 800 different species, so the real question is to ask, is the microbiome in totality, what are they doing? And so there's certain neurotransmitters that are being produced by the microbiome, including things like serotonin and GABA, and things that influence our mood, our anxiety, our sleep, and so forth. And one of the things that my laboratory studies is not only breast milk, is cannabinoids. Who here knows what cannabinoids are? Raise your hand. These are the active ingredients in marijuana. And so when you take an edible, when you chew that edible with cannabinoids, how does that modify the microbiome to perhaps produce a different set of neurotransmitters? And so there's active research about okay, certain microbiomes produce certain neurotransmitters. How can we impact that with probiotics, prebiotics, dietary interventions, lifestyle interventions, that kind of stuff?
Okay, we have one here.
Out of all the available microbes, what led you to choose to study bifidobacteria?
Of all, there are so many microbes. Okay, this started before me and will end after me at one point in time. There was an observation that occurred 100 years ago. And this is, scientists who are working at Nestle, I think, that were looking at formula-fed babies versus breastfed babies. And the breastfed babies had a ton of bifidobacteria. And so someone said along the way, I think bifidobacteria is important without any evidence. Very similar to the urea thing. We're very curious, like, urea is there, like it must be useful for something. Let's take a look at that too. And then only recently, in the last 20, 25 years, we had the tools to study bifidobacteria to say, yeah, it is important because it does all those things that I showed on that list. But if you're asking me, is bifidobacteria the most important? It's the most important to me right now. But I'm curious about how they interact with their near neighbors. And how do they interact with a group of 10 different bugs? And how do they interact in the what's known as the emergent properties of the microbiome as a whole? Good question.
Okay, this might be our final question.
This is an easy one. Probiotics, yay or nay?
Ooh, you thought it was going to be an easy question. This is my take home for that. Depends on the strain, depends on the person, depends on the condition. And so how do you actually, if you happen to be working in the clinic, so how can you advise and guide people? Well, there's actually no good rubric for it. So probiotics, the best, and this goes back to, as you said, vaginal seeding, and this goes back to fecal microbiome transplants. We know that there's ways that we can add back beneficial bacteria that make a difference, that help people out. So we know the probiotic concept works. We cannot predict very well right now if a specific strain does anything for a person without saying, try it. And if those of you who are taking probiotics, you might say, oh, I see a reduction in gas and bloating. And then why not keep on going on with that probiotic? And I don't want to diminish the impact of the placebo effect, but that's something that's actually very powerful. Our minds are much more powerful than we actually thought they were too, which I'm not saying that you should delude yourself into thinking, I'm gonna take this probiotic now, I'm gonna be cured of cancer. That's actually not the case. But if that probiotic you're taking does something for you, why not keep on taking it too? It's not snake oil. So I'm gonna say a very guarded, yes, thumbs up on it. But in terms of being able to measure it in all instances, we can't measure it in all instances. Well, depending on the probiotics, they may not be naturally predisposed to, it's known as engraftment. So to stick around for a while there too. You also have your native microbiome there too that doesn't want to give up its territory, its physical location as well as access to food, and it's a stable community too. It's sort of like if you get a packet of seeds at a store for flowers and you go to a forest and you spread those seeds out, it's not like those seeds are going to just clear out the forest. It's the same thing with probiotics. They're not going to clear out what you have present there, and so you're just going to have them continuously pass through you. Sometimes they stick around for longer. If you happen to have certain conditions, it would be wise to take probiotics. Necrotizing enterocolitis, there's a big, there used to be a big scientific debate, and there's a lot more evidence for it now that probiotics are helpful with necrotizing enterocolitis, something like that. So it just really depends.
Okay, one more, just because. So could you change your eating habits to make a probiotic flourish and last longer so you don't have to take it every day? Yeah.
Absolutely. So so the question I would have for you, are you part of a scientific study where we're going to measure that, or are you just going to do it based on the way you feel? So if it's going to be based on the way you feel, if, let's say you're trying to treat excess gas and bloating, something like that, and you're taking a probiotic, but you're also changing other elements of your lifestyle, it might be very difficult to decouple that, what's the impact of the probiotic versus, well, now I'm running at least a mile or two a day, and I'm feeling better about myself. So, and then if you go to a clinician, you go to a scientist, you go to someone else, like, should I continue my probiotic or should I continue running? Do both, right?
Let's thank the speaker. We'll thank him a couple more times. Yeah. A great talk. So I'm actually going to send the speaker away for a minute or two. If you can select 2 questions from the list for best question prizes, you can leave the mic there. We do have some fabulous prizes. So we have a Bifido, giant microbes art mug, and a bacteria science kit. Ooh, whoa, well, ages 9+. So we're going to do the door prize selection first, but the rule is if you get the door prize, you have to select it last. You get last choice, okay? Incentivizing questions. I'm just gonna pick on audience member here. So your blue ticket must be present to win. Yeah, drum roll. All right, so we have, last 3 digits are 582. Do I have 582? Do we get a woo? Woo! All right, so unfortunately you get last choice, but there will be a prize for you. And so what are the 2 best questions? How could this improve infant health? Does anybody remember this one? Right, woo! Okay. And Eliza's question. Eliza also gets to select a prize. Yay, Eliza, thank you. We're not quite done, so come get your prizes afterwards. To thank the speaker, we have a lovely parting gift for him. Otherwise, make sure you're signed up for our mailing list. You can go to scitechcafe.org and click on some links to get on the mailing list. We'll send out the dates for the next season, which will start in the fall. Hopefully have some speakers to announce, and we'll be back here in September. Thank you so much, drive home safe, and let's thank the speaker one more time. And as one final reminder, please bust your tables, please buy a few more beers from the brewery and tip your bartenders, and thank you so much.